Chapter 2 of Virology, the infectious cycle. Today we're going to talk about what viruses do once they get into cells. And we're going to consider some general principles as well as some methods for studying the infectious cycle. And all of these will be some things we come back on throughout this course. So it's important that you get familiar with this material. So the infectious cycle is what happens when a virus encounters a cell. It's all the various steps that a virus undergoes in order, in the end, to produce more infectious particles. As you can see here on this diagram of a cell is illustrated the reproductive cycle or the infectious cycle, the terms are synonymous, of a particular virus. And the red arrows highlight some of the specific steps in infection. So all viruses need to attach to cell receptors. That's step 1 here. The virus particle sometimes gets into a cell, sometimes it's just a genome. But in either case, the nucleic acid genome of the virus has to get into the cell. Here it's shown going in the cytoplasm. And you'll see for other viruses that it may end up in the nucleus instead. The genome has to be made into protein, the process of translation. The genome has to be replicated, and that's shown here in a big complex scheme occurring on membranous vesicles, making more genomes from the input genome. Those new genomes, shown here, then have to be packaged into new virus particles. That happens when the proteins that encode, or that are the viral capsid proteins, are made, get assembled into a new capsid and coalesce around the viral genomes. And finally, these new virus particles have to get out of the cell. And in this case, they are shown escaping when the cell lyses. So these are the various steps of the infectious cycle, and they differ for different families of viruses, depending on the structure of the virus, depending on the nucleic acid of the virus, and other properties as well. And these are considerations that we'll go into in the remainder of this course. So that's what we mean by the infectious cycle, everything that happens from start to finish as a virus is replicating, as a virus is producing new infectious particles within the cell, because remember, all viruses are obligate intracellular parasites. Now, we divide the infectious cycle into steps, as I've shown here with these red arrows and the numbers. Of course, there are no steps within a cell. Everything happens in a continuum. There's a beginning when the virus attaches, and there is an end when the virus lyses. But many of these things happen at the same time. We make these divisions in order to make it easier for us to study them. And in a course like this, it's easier to explain by dividing the cycle into steps. In fact, this course, at least the first half of this course, every lecture is based on a different step in the infectious cycle. After a few introductory lectures, we'll start going through this infectious cycle one step at a time. And that's a highly artificial procedure, of course, but it's great for teaching you how viruses work. Now, we need to put some definitions up very early on because I use these terms over and over again in this course, and you need to be familiar with them. First, the term susceptible as applied to a cell. When I talk about a susceptible cell, I'm talking about a cell that has a receptor for a particular virus. If I talk about cells susceptible to poliovirus, I'm talking about cells that have a receptor for that virus. Cells susceptible to influenza virus have a receptor for that virus, and so on. Whether anything else can happen when that virus attaches to that cell is not implied by susceptible. So that's why I say here the cell may or may not be able to support virus replication. All susceptible means that there is a receptor for the virus on the cell surface. A resistant cell, in contrast, has no receptor. That doesn't mean that that cell could not support virus replication. You can imagine that we could put the nucleic acid of the virus directly into the cell and determine whether that cell could support replication or not. So resistance simply applies to having a receptor or not. A permissive cell has the capacity to replicate virus. It may or may not have a receptor for that virus. In other words, a permissive cell, you could introduce the nucleic acid of the virus and it might replicate, but that cell may not have receptor. Finally, a susceptible and permissive cell is the only cell that can take up a virus particle and replicate it. So those are two specific definitions, susceptible and permissive as applied to virus infection. And many virologists confuse the terms. I don't want you to confuse them. I want you to know what they mean because it makes teaching and learning about viruses that much easier when you know the definitions. Now, way back when viruses were first discovered, we talked last time about how viruses were discovered around the beginning of the 1900s. For a long time, the animal viruses, which are the main subjects of this course, couldn't be propagated in cells in culture. This is a technique that we all use today extensively to study virus replication. But in the beginning of virology, viruses had to be propagated in laboratory animals. And some of the common animals used for virus preparation are shown here. The mouse is a very, very commonly used animal for a number of reasons. It's relatively small and easy to breed. And the mice can be inbred so that the strains of mice are homogeneous. But other animals that are not inbred can also be used. Chickens played a large role in our understanding of tumors caused by viruses, as we'll see later in this course. Non-human primates are used to study viruses, as well as monkeys and other animals such as cats, dogs, and hamsters. One of the more commonly used non-cell culture hosts for a virus is the embryonated chicken egg, which is diagrammed here. So this is the kind of egg that you would have yourself for breakfast, except that it's embryonated. It has a developing embryo in it, as you can see in the center. And for influenza virus, which is one of the viruses that can be propagated in eggs, the eggs are taken at 10 to 11 days post-fertilization. On the right here, you can see all the different kinds of viruses that can be grown in different parts of the egg. Depending on where you inoculate virus into the egg, shown by these needles, you can grow different kinds of viruses. And there are lots of different kinds of cell types in the embryonated egg, and that's where the viruses replicate. So for example, a common place is to inoculate influenza virus into the allantoic cavity, which is this large cavity here. It's filled with blue color surrounding the embryo, and the virus is inoculated in there. You drill a hole in the shell, you put a needle in, you put virus in. The virus will replicate on the cells lining the cavity. And then after a number of days, you can withdraw the allantoic fluid, and you have 5 to 10 mLs of virus preparation, so it's very convenient. Now this approach using embryonated chicken eggs has largely been superseded by using cell cultures, except in one major case, that is influenza virus vaccine production. Embryonated chicken eggs are used to produce influenza virus vaccine. There are certainly other ways you can produce influenza virus vaccine as well, but this is still one of the major ways. And one of the reasons why is that it's relatively easy. There are machines that automate the inoculation process for each egg, and you can get 10 mLs of virus per egg, and that gives you about 1 dose of vaccine. So the next time you get your flu shot, you had actually a couple of eggs worth of flu viruses going into your arm because there's more than one virus in the vaccine. However, at some point in the history of virology, we moved towards cell culture, away from animals and embryonated chicken eggs. And that development was spurred by this virologist, John Enders. In 1949, he and two of his colleagues, Weller and Robbins, showed that they could propagate poliovirus in a human cell culture. In this case, they used primary cultures of embryonic tissues. This had never been done before. By this time, scientists had figured out how to grow cells in culture, and Enders, Weller, and Robbins figured out how to use them to grow viruses. This was a big deal, a big discovery. They got the Nobel Prize for this discovery in 1954, And as you can see, John Enders made it on the cover of Time magazine, which has happened only a few times in the history of virology. So this discovery facilitated the study of viruses for the next 50 years, and indeed to this day continues to be used to study viruses. In addition, in the 1950s, this discovery facilitated the development of the first polio vaccine. Today we grow viruses in several different kinds of cells that are grown in the laboratory on plastic dishes. And here are 3 different kinds of cells. They have distinctly different properties. So on the left is a primary human foreskin fibroblast. This kind of cell culture is made from fresh tissues, in this case foreskins, which are very easy to obtain if you're at a medical center because they're generated on a daily basis. You mince them up, digest them with trypsin, and put them on a plastic dish, and the cells adhere, and eventually they will divide and make a nice monolayer, as we call it, and you can see that here. And these can be infected with a variety of viruses. The disadvantage of these primary cultures, not just foreskins, but other tissues as well, is that they don't live forever. After 20 to 30 doublings, they will die, and you have to make them again. So there are obviously problems with that, so therefore continuous cell lines have been developed. And here are shown 2 of them. One is a mouse fibroblast cell line called 3T3, and the one on the right is a human epithelial cell line called HeLa cells. And these are cells that have been propagated in culture. They actually have been started from a primary culture, and then in various ways they're immortalized, which makes them live forever. In the case of the HeLa cell line, these were derived from a tumor, a human tumor, and that in itself made them immortal, and they grow forever. And why that is so, we'll talk about later in this course. So these 2, the 3T3 and the HeLa, are called continuous cell lines. The problem with those, they last forever and they're very convenient, but the problem is that they're aberrant. They have abnormal numbers of chromosomes. They're not diploid by any means. And if you want your cells to be normal for your virus studies, there is an alternative. There are diploid cell strains, for example. One of them, the famous ones, is called WI38, which is derived from a human embryonic lung. So there are all sorts of options to use when you study viruses in cell culture, and often you can pick a cell line that's derived from the tissue in which your viruses normally replicate in the host. In our lab, we use lots of different cell lines to propagate viruses in different ways. And here you can see that the cells are grown in plastic dishes, and they're covered with medium, which provides nutrients. We use dishes of various sizes as well as flasks. And they're all incubated at 37 degrees Celsius in a 5% carbon dioxide atmosphere to buffer the medium so that as the cells produce metabolites, the pH is kept constant. We also use HeLa cells, which I mentioned before, and we grow these in what we call spinner cultures. We do this because we need a lot of these cells, and a spinner culture is the best way to do it. Here we have a liter of medium in which the cells are growing, and in this flask, which is of course sterile, it's kept in a room that's 37 degrees Celsius, there's a magnet, a suspended magnet on a glass rod, and that magnet turns by virtue of a corresponding magnet in this plate beneath it, which is run by a motor. So this spins continuously, the cells divide every 24 hours, and we can use them for our virus experiments. Now in my lab, we study polioviruses and rhinoviruses, and we use a lot of cells. And we use a lot of HeLa cells. And if you're interested in the life or the history of HeLa cells, this is a great book to read, The Immortal Life of Henrietta Lacks by Rebecca Skloot. And HeLa cells were taken from Henrietta Lacks in 1951. But she died shortly thereafter, and they were the first cell line, the first human cell line to grow forever. They are immortal, and they're still growing in many laboratories today. But her family didn't know about this discovery, and when they found out, it was quite disconcerting. So this book is the story of the family, essentially, in that cell line. I highly recommend it. Now, we don't have the option of doing a Socrative today because thanks to the blizzard of historic proportions, we have no class today, and this is pre-recorded. So I'll give you a moment to look at this question and try and answer it. The question is, a blank and blank cell is the only cell that can take up a virus particle and replicate it. So you need to fill in the blanks. And the options are 1, naive and resistant, primary and permissive, susceptible and permissive, susceptible and naive, and continuous and immortal. So which two best fit that sentence? If you would like, you can pause the video at this point because I'm going to give you the answer right now if you need some more time to think of it. The answer, of course, is number 3, susceptible and permissive. Remember, susceptible cells have a receptor. Permissive cells mean the cells can support virus replication. That means everything that goes on past the receptor step. So now we're at the point in the history of virology where we have cell cultures, such as the one in the upper left, that can support virus replication. So let's go through a number of ways that we can use these cell cultures and what we can learn from them. And the first is, how would you know that your virus is actually replicating in a cell culture? So we take these cells in the upper left and we infect them with a virus. It happens to be poliovirus in this case. Well, one way to know is by what the virus does to the cells. And if you track from the upper left to the panel on the upper right, then to the lower left and the lower right, you can obviously see changes in the cell culture are taking place. These are 4 photographs of the same culture taken at different times after infection with polio. This is 0 hours, this is 2 hours, this is 4 hours, and this is 8 hours. So you can see that the cells are rounding up and detaching from the monolayer. And these changes, this is one kind of such change, these changes are called cytopathic effects, or CPE. And I'll be talking about this quite a bit throughout this course, so let's delve into this a little bit. CPE simply means that the changes in a cell that you can see that are caused by virus infection. So the detachment and rounding up of cells that I just showed you is one example. Another example of CPE, or cytopathic effect, is shown here, and this is the formation of syncytia. Now, a syncytia, or that's the plural, or syncytium, which is singular, is nothing more than a number of cells that have fused to become one giant cell, as shown in this illustration at the right. So here we have 2 cells fusing, and then they become one bigger cell with 2 nuclei, and that can then fuse with other cells, and eventually you get a big cell or a giant cell with many nuclei. That is called a syncytium. And on the left is a photograph of an actual syncytium observed in cell cultures. You can see individual cells and syncytia. And this is a culture that's been infected with measles virus, which causes the cells to fuse. And you can see these large cells, the syncytia, have many nuclei in them. One is pointed out by this arrow. Why does this happen? Well, as we'll see later when we talk about the replication cycle in more detail, in many virus-infected cells, the virus replicative cycle results in the production of a receptor binding surface on the surface of, a receptor binding protein on the surface of the cell, and that's shown by the red Y. The function of that, of course, is to engage in binding to a cell surface receptor. And this is placed on the surface because the way these viruses are produced is by budding, as we'll see later. It's the first step towards making a new virus particle. Well, even before a new virus is made, this cell is able to bind to a ligand for the receptor on a neighboring cell, and that act often leads to fusion. So this is sort of a byproduct of the replication of viruses in cells, but it contributes to cell death, and it certainly is another form of cytopathic effect. Here's a list of many different kinds of cytopathic effects that we see in animal cells infected with different viruses. So the effect is shown on the left, and the virus is shown on the right. And I show this to you just to give you an example of the wide range of alterations that can happen. For example, we have one class called morphological alterations. We have nuclear shrinking, pycnosis, or proliferation of the membrane of the nucleus, production of vacuoles in the cytoplasm, syncytium formation, which we just talked about, the alterations in chromosomes, rounding up and detachment of cultured cells, which I illustrated for you previously. And then another class of cytopathic effects comprises the production of what are called inclusion bodies. These are bodies within the cell that aren't there in the uninfected cell, but which are caused by virus infection. And they include actually virus particles in the nucleus or in the cytoplasm. And these are given names sometimes because years ago when they were first observed by a pathologist, for example, he or she would say, aha, look at this is unusual and is typical of rabies virus, so this was called a Negri body. But it's nothing more than clumps of virions in the cytoplasm. There could be factories in the cytoplasm. These are assembly sites for virus particles. This happens with pox viruses and a few other viruses, and those have been called Guarnieri bodies. You can also have clumps of ribosomes and virions, not really a cytopathic effect, but is an inclusion body, so it's on this slide. And you can have clumps of chromatin in the nucleus. So there are many ways to know how your cell culture, or if your cell culture is infected, simply by looking at them. And all of these are apparent under a light microscope, which is rather low power. You can't see virus particles in a light microscope, but you can certainly see the effects that they're causing on cells. The next question that we have, if we can grow viruses in cell culture, we need to know how many viruses there are in a sample, either a sample that we're adding to the cells, or if we're harvesting virus from infected cells. We have to be able to count the number of particles or quantitate the infectivity. And there are two ways to do this. As I've just said, infectivity is one. We can measure infectivity, but sometimes we can't do that, in which case we have physical assays for counting the number of particles. We can count the particles in a variety of ways, or certain components of the virus particles, and that'll become clear in a moment. So let's first talk about infectivity. The first assay, and perhaps the most powerful, is the Plaque assay. And it's called that because the virus is forming plaques on the monolayer of cells. On this slide, I'm showing you a Plaque assay with a bacteriophage, a virus that infects bacteria. First, the Plaque assay was first developed using bacteriophages in the 1930s. And what we have here is an agar plate. It's a petri dish, a plastic petri dish with an agar layer on it. And on top of the agar, this is looking from the bottom of the plate, was spread a monolayer of bacteria. And that monolayer had been mixed, the bacteria had been mixed with viruses prior to putting on the plate and incubated, and wherever, and the bacteria form a very nice monolayer, which is shown by the cloudy nature of the color. And then wherever a virus is replicating and killing the bacterial cells, you have a clearing. You can see many of them here. And that is what is called a plaque. So plaques were actually first discovered with bacteriophages. But then later, the Plaque assay, which is a way to measure the number of plaques, was then extended to animal viruses. This was done in 1952 by the virologist Renato Dulbecco. And he took the Plaque assay developed for bacteriophages and made it work for animal viruses. And on the upper left is one of his early Plaque assays. Now, he received the Nobel Prize for this and other discoveries in 1975. Here is the abstract and title of his paper, which was presented in April of 1952, Production of Plaques in Monolayer Tissue Cultures by Single Particles of an Animal Virus. This was a big deal. We use the Plaque assay to this day to measure and quantify virus growth. Now, notice he says single particles of an animal virus. I'm going to show you in a bit how he knew that the plaques were actually formed by single particles. First, here's how a Plaque assay works. Let's say we have a tube of virus stock. That's the one on the left here. And we want to know how many infectious viruses are in this tube. So then you make a series of dilutions. You take a number of tubes, you put 0.9 ml of an appropriate buffer in them, And then you do tenfold serial dilutions. You take 0.1 ml of the virus stock, you put it into the first tube, mix it up, take out 0.1 ml, put it into the second tube, mix it up, and so on down the line. And so these are the dilution factors that you then get. You then take some of the later dilutions and you do a plaque assay with them. You infect a monolayer of cells with 0.1 ml in this case. You take off the medium from the cells, you do the infection, and after you let the virus attach to cells, you add an agar overlay over the cells, which solidifies, and then you put the cells in the incubator. And then after a few days or a week or two, depending on the virus, you can remove the agar overlay and stain the monolayer, and you can see your plaques formed. And then you can count them. So here, for example, we have too many plaques to count on the left. We have 17 in the middle, and then we have 2 here. And what you'd like to pick is a number between 10 and 100, which gives you a reliable count. From this, we can calculate the plaque titer. So we have 17 plaques on the dilution of 10 to the minus 6. So that's 17 times 10 to the minus 6, or 1.7 times 10 to the minus 7. I move the decimal point over 1, and we go up to minus 7 here. But that's not the answer that I'm showing here. It's 1.7 times 10 to the 8th, and that is because we used only 0.1 ml to plate out here, so we have to add another tenfold to the dilution factor. So the titer of this virus is 1.7 times 10 to the 8th plaque-forming units, or PFU, per ml. So it's very important when you calculate the plaque titer to take into consideration not only the dilution factor, but also the additional factor imposed by plating only 0.1 ml. If we had plated a mil, then we wouldn't have to add that extra dilution factor here. So that's the plaque assay. And here's a diagrammatic representation of it above. Again, we have a monolayer of cells. We infect with a dilution of virus. We cover the monolayer with an agar overlay. It's a solid overlay that prevents the virus from spreading through the culture once it's released. So let's say in this case the virus has infected a single cell in this section of the monolayer. That cell will eventually break open and release virus particles. And because the cells are covered with an agar overlay, those particles are only restricted to the neighboring cells. They can't spread throughout the culture. So the neighboring cells are infected and they lyse, and then those viruses infect neighboring cells. And so you can see this zone of dead cells simply gets larger and larger until it's visible by the naked eye, and then it's a plaque. If we didn't have an agar overlay on this cell culture, the viruses produced by this first cell, or the first cells infected in the entire plate, would spread throughout the culture, and they'd lyse every cell by the second or third round, and you'd never see any plaques. So we use an agar overlay to restrict diffusion of the virus in the cell culture. On the lower left is a light micrograph of a single plaque, just to show you what a plaque looks like in real life. The top was a diagram, of course. So here you can see a monolayer of cells, and in the middle the cells have been rounded up and they're dead. They're detached. When you take the agar overlay off of these cells will wash away, and that will be your plaque there. And on the right is a plaque formed and visualized in a slightly different way. This is a virus in which we've inserted a gene encoding an enzyme that will convert a chemical to a blue substrate. So after letting the virus infect cells for a while, we can stain the monolayer with the substrate. And you can see this is a plaque formed by the virus. There's a little bit of cytopathic effect in the middle, but you can detect the plaque very easily by the blue color, which is again made by the enzyme produced by the virus. Now this is a movie which displays plaque formation. I'm going to stop at the very beginning, just to explain to you how it works. So this is a time-lapse movie where a monolayer of cells, which you can see here, were infected with a virus, and then after a few hours, a single forming plaque was identified. So here's the very beginning of a plaque with just a few cells infected. There's a little hole in the monolayer here. Then they put a camera on this single plaque and took a frame every 15 minutes or so, say. And then they put all the frames together so we have a time-lapse photograph. So what you're going to be able to see is the plaque growing in real time. So let's see how that works. You can see the plaque hole is getting bigger and bigger. You can see the refractory change of the cells as they get infected and detach from the monolayer. So they become shiny, if you will, as opposed to dark, which they are uninfected. And you can see the time here, 8, 9, 10, 11, 12, 13, 14, 15 hours it took for that plaque to form. So this was the first movie ever made of a plaque, actually in real time. It happens to be vaccinia virus, which is a pox virus infecting cells. I find this mesmerizing. I think this is one of the greatest movies ever produced. I really think the plaque assay is something special. We do lots of plaque assays in my laboratory, and here is a wall made up of plates that we've kept. You can see this is a 6-well plate, and maybe you can just see the plaques on this plate. They've been stained with a dye called crystal violet, and you can see the tenfold dilutions. There are a few plaques here, in the middle there are more, and there are even more on the left. And these are all plaque assays to assay poliovirus, and we call this the wall of polio. This used to be standing in my laboratory, and I took many pictures of famous people in front of it, as well as students from prior versions of this class. You can see all those portraits here at this website. Unfortunately, the wall of polio has fallen a few times. I'm presently in the process of reconstructing it in my office. So if you come for office hours sometime, you'll see it. Let's try another question now. When doing a plaque assay, what is the purpose of adding a semi-solid agar overlay on the monolayer of infected cells? And the options are 1, to stabilize progeny virions. Number 2, to ensure that cells remain susceptible and permissive. 3, to act as a pH indicator. 4, to keep cells adherent to the plate during infection. Or number 5, to restrict viral diffusion after lysis of infected cells. I'll give you a few minutes. You might want to pause your video right now and answer it, because I'm going to give you the answer now. The answer, of course, is number 5. To restrict viral diffusion after lysis of infected cells. If you didn't do that, you would never get a plaque formed. So here is an example of a very nice plaque assay. Again, it's stained with crystal violet. You can see the dilution series going from right to left. And I would count the plaques in the middle. The one on the left has too many. You can see they begin to overlap each other, and you can't really count them reliably. This happens to be a plaque assay with influenza virus. Now, back when I told you that Renato Dubecco had developed the plaque assay for animal viruses, in the title of his paper it said one virus forms a plaque. So let's figure out how he figured out how many viruses are needed to form a plaque. And the experiment to do this is pretty simple. You just do a dose-response curve. You make dilutions of virus, which are plotted on the x-axis. You do a plaque assay, and then you count the number of plaques, and you plot them on the y-axis. So here you have increasing concentrations of virus, and you simply plot the two numbers versus each other on a simple graph. If one virus particle is sufficient to form a plaque, the relationship between the virus concentration and the number of plaques is a straight line, as shown in red. And that's because if a single particle is sufficient, that's one-hit kinetics. The number of plaques is directly proportional to the first power of the concentration of the viruses inoculated. And all that means is simply you have a straight line, because as you double the concentration of virus, you double the number of plaques. So you go from 0.25 to 0.5, and you go from 6 to 12 plaques. Sometimes you need more than one virus to form a plaque, and that is called two-hit kinetics. In this case, the number of plaques is directly proportional to the square of the concentration of the virus inoculated, and you get a curved line such as this. Some viruses do have two-hit kinetics because they are comprised of two pieces of genome, and they're packaged in separate particles. But for most of the viruses we'll be talking about, they have one-hit kinetics, and Renato Dubecco simply did an experiment like this to prove that a plaque was formed by one virus particle. Now this is important because if you would like to purify stocks of viruses, you use a plaque assay, you want to make sure that just one virus is causing that plaque to form, and that's in fact the case. So we use plaque purification to produce clonal virus stocks in the laboratory. You do a plaque assay, and each plaque is initiated by a single virus particle, as I've just told you, as proven by the dose-response curve. You can then take a pipette, you plunge it into the agar over this plaque. So you pick up a plug of agar, and then you put that plug of agar into some buffer, and the virus in the agar plug will diffuse, and now you have what we call plaque-purified virus stocks. We usually repeat this process 3 times because you're never sure if this is actually formed. One plaque that's forming here, there could be a smaller plaque that formed coincidentally in the same area. So you do it 3 times just to make sure that you have clonal stocks. So that's the plaque assay. Another way to titrate virus infectivity is by what's called an endpoint dilution assay. Some viruses do not form plaques. So what can you do? You need to find something else, and that's what this assay is for. You take a 96 well plate with 96 little wells in it, and you put your cells in them to form monolayers. You have a little bit of medium over each one. You infect each well with a different dilution. The first row is infected with a -2 dilution, then -3, -4, and you go all the way out as far as the 96 well will handle. Then you incubate the plate for a few days, and you look at each well under the microscope, and you simply ask whether you see cytopathic effect, which is evidence that virus infection has occurred. So you can see here the results are plotted on this table. On the first row is the -2 dilution. There's a lot of virus here. This is not much of a dilution, 1 to 100. And you see cytopathic effects in all of the wells. -3, same thing. Now in -4, we have one well where there's no cytopathic effect. So now we're diluting it sufficiently so that just by chance some wells are not going to get any virus. You can see that in -5, there are actually an equal number of plates with and without cytopathic effect, and then as we dilute further we get less and less cytopathic effect. In this assay we'd like to find the dilution at which 50% of the wells are infected, as evidenced by the production of cytopathic effect. And that number will be our way of quantifying the amount of virus. We call that the TCID50, tissue culture infectious dose 50%, how much virus you need to infect 50% of the wells. And for this virus stock, it happens to fall on the 10 to the -5 dilution. It doesn't always fall right on a dilution, but you can use math to extrapolate interdilution amounts. So here we would say that this virus stock contains 10 to the 5 or 100,000 TCID50s, and that's a kind of quantification that you'll see quite often in the virology literature. Another important aspect of virology is the particle-to-PFU ratio. This is a very simple concept. It's simply the number of physical particles divided by the number of infectious particles. So those are 2 different measurements. You have to be able to take a stock of virus, count the physical particles, and we'll talk about how we can do that in a moment, and then measure the infectious particles, which you could do by a plaque assay or a TCID50, as we've just discussed. This number varies from virus to virus. For some viruses it's 1, for some it's 100, for some it's 10,000. So a particle-to-PFU ratio of 10,000 means that there's only 1 infectious particle for every 10,000 physical particles. In other words, not every virus particle is apparently able to complete infection. Now, from what I've told you just previously, showing you the dilution series, showing that one virus particle can initiate an infection, we know that in theory, any virus particle can initiate an infection. But for some reason, not all of them do. And that's reflected by the particle-to-PFU ratio. So not all viruses are successful at initiating, even though in theory 1 virus is enough, as shown by the dose-response curve. Why? This has been a problem that has puzzled virologists for years. I have some possible explanations here. Whenever you grow up a virus preparation, sometimes the virus particles can be damaged in the prep. There may be mutations in the genome, or just randomly, because a virus has to go through so many steps in an infectious cycle, if just one fails, it's not going to make it. And so that could explain this as well. Now, for the most part, the particle-to-PFU ratio is more or less constant for a given virus. So, for example, for poliovirus, it's typically 1 or 2 or 300 particles to 1. And for some phages, it's 1. This can vary a little bit as you prepare your virus, but it's usually pretty constant within the virus preparation, or the virus family, I should say. Why am I telling you this? Well, this can complicate the study of viruses, because if you have a particle-to-PFU ratio, say, of 10,000 to 1, and let's say you're infecting cells with a virus and looking at it in the electron microscope, how do you know which particles are actually moving along the infectious cycle? You don't know, because if 10,000 are non-infectious, that is really a problem. So people have tried to work with viruses that have particle-to-PFU ratios that are as close to 1 as possible. Here are some particle-to-PFU ratios of some animal viruses. Among the best are the alpha viruses, Semliki forest virus, in which almost every particle is infectious. And you can see that papillomaviruses are terrible. Only 1 in 10,000 are infectious. And then you have a range of values for other viruses as well. Something to keep in mind whenever you're studying viruses and looking at physical aspects of virus infection. All right, let's take a pause and have a question. In the property known as particle-to-PFU ratio, particle can best be described as, and the possible answers are 1, one of the proteins which makes up the virion; 2, a virus which may or may not be infectious; 3, a virus which is infectious; 4, a virus which is not infectious or may be a virion. And you might want to pause the video at this point, because I'm going to give you the answer. The answer is a virus which may or may not be infectious. The particle makes no implication about infectivity. It's just a physical virus particle, and the ratio of that to infectious particles is what the particle-to-PFU ratio is. Because when you count particles, for example, by electron microscopy, you have no way of knowing if they're infectious or not, and that's simply what a particle is. Okay, now we have some methods of measuring viruses and telling when cells are infected. What can we do with that? Well, one of the most important techniques in virology is the one-step growth cycle. It's a way of studying that infectious cycle, which gives the name to this lecture, in cell cultures. And this was first developed in 1939 by 2 virologists, Ellis and Delbruck, who were studying phages that infect Escherichia coli. And what they did was to develop this one-step growth cycle to study the replication of phages in cells. And the procedure is done as follows. You take your phage and you add it to the bacterium, and you allow it to adsorb. This is a word that simply means the virus is attaching to the bacterial cell. You let it absorb for a period of time, typically 30 to 60 minutes. Then you dilute the culture so that there's no more adsorption. It's now so dilute that the likelihood that a virus is going to impact on the bacteria is very low. You want to synchronize infection. Then you let the infection proceed. You sample at various times, and then you measure virus infectivity. It's a very simple experiment, and it tells you a lot. So here's an example of a one-step growth cycle on the left. We're looking at the x-axis at time, and on the y-axis, the number of infectious particles. So remember, we've allowed the viruses to adsorb to the bacteria, and then here at this first arrow, labeled with start dilute, we dilute the culture, and that begins the experiment. We start taking time points and measuring virus infectivity. You can see for a while, the while can be 10 or 20 minutes or an hour, it depends on the virus. There's no infectivity. And then all of a sudden, there's a steep increase in virus particle production, and eventually it plateaus. And the actual time on the x-axis really depends on the virus. If it's a bacterial virus, it can be relatively rapid. What's going on here? Well, here in the eclipse period, the virus is attached to cells. It's put its nucleic acid into the cell, and that nucleic acid is directing the synthesis of viral proteins and eventually new virus particles. Remember, viruses don't divide by binary fission. If we added a bacterium to medium, it'd start growing right away. We showed that slide the other day. But viruses don't do that. So there's an eclipse period, which is defined as simply the time during infection when new virus particles are made, but they're not made yet. And then when you first detect infectivity in the experiment, that's when the new virus particles are completely assembled and now they're infectious. They can cause an infection on, say, a plaque assay. Eventually you exhaust the culture, you kill all the bacteria, and so no virus particles can be made. So the two terms here are the eclipse period and the burst or the yield. That's a single-step growth cycle. I think you can see why it's called single-step, right? Because there's one burst of virus yield and that's it. The reason it is single-step is because we've infected every cell in the culture. What if you infect a few cells to begin with? Not all of them. Say you infect in a 1,000,000-cell culture 10 cells. Well, you do the same experiment. You start the experiment by dilution, you measure your time points, you see you have an eclipse period, then you have a burst, and then you have a plateau, and then you have a second burst and a plateau. And this can go on depending on how few cells you initially infected. What's happened here is the few initial cells infected produce virus, and they go on and infect other cells, but there's another eclipse period at which time they're producing new particles in those other cells, and then a second burst, and so on. So this is called a multi-step growth cycle on the right here. Both single and multi-step growth cycles have different applications, as we'll see in virology. Here's an example of a single-step growth experiment for an animal virus. Here we've taken adenovirus type 5, which is a rather large DNA-containing viruses, and we have infected mammalian cells in culture. Same principle. You infect all of the cells in the culture, you adsorb the virus, then you dilute to prevent any more adsorptions, and then you take time points at different times after infection. And you can see here that there's an eclipse period as well, during which no infectivity is detected. And then at about 12 hours after infection with adenovirus, you start to see the production of viruses. And eventually, the virus production plateaus as the culture is basically killed and no more viruses can be produced. So similar terminologies. We have the eclipse period here, and here is the yield, of course. But one additional term is shown in this slide, and that's because we've measured viruses in a different way from the previous experiment. Here we've measured both intracellular and extracellular viruses. So extracellular viruses are easy to measure. We simply take the medium from the cells and do a plaque assay. And to measure intracellular viruses, we have to actually take the cells off the dish, break them open, and do a plaque assay on the contents of the cells. So you obviously have to do replicate cultures in order to do both intracellular and extracellular viruses. When you plot the extracellular virus production, you see an interesting fact. You don't see viruses in the medium until 16 hours after infection. But I told you at 12 hours, the particles are made intracellularly. So there's a 4-hour lag between the time the first particles are made and the time that they get out into the medium. That's called the latent period, simply the time between when the viruses are made and when they're released from the cell. So it takes time for the virus particles to get out. So now we have eclipse period, we have latent period, and the yield or the burst. And remember, bacteria grow in a very different manner. When bacteria are introduced in a culture, they immediately begin to divide, and this is completely different from viruses because bacteria divide by binary fission. And remember, in a virus-infected cell, there's an eclipse period because it's during this time that new virus particles are assembled. They do not divide by binary fission. You take one virus and you make many eventually, but one bacterium divides into 2, 4, 8, etc. Now, to do a one-step growth curve, only one step, not multi-step, you have to infect all the cells. How do we know that we've got all the cells infected? Now, I told you how you can measure virus infectivity, say, by a Plaque assay. But if I gave you a plate of 1,000,000 cells and I said, put enough virus on this to infect all of them, how much would you add? Well, the answer lies in another term that we have to remember, the multiplicity of infection, or MOI. We're going to see this throughout this course, so you need to learn this. Multiplicity of infection is defined as the number of infectious particles added per cell. It's what you add as the investigator to the cell culture. It's not the number of particles that each cell will end up receiving. That is actually quite different, and we'll talk about that in just a few seconds. So, an example, if I take 10 million 10 to the 7th virus particles and add them to a culture of 1,000,000 cells, that's an MOI of 10, because each cell is getting 10 virus particles. Okay, that's the definition of MOI. However, each cell does not receive 10 virus particles. All right, so that's the difference between MOI, which is what the investigator adds to the cell, and what the cell actually received in terms of the number of virus particles. So why are the terms so different? The reason is that when you infect cells with a virus, the infection of a particular cell depends on random events, random collisions of viruses and cells. And so that gives you a range of events. So if you take susceptible cells, remember, that have receptors, and you mix them with virus, some cells will bind 0 virus particles, some will get 1, some will get 2, some will get 3, some will get more. This is characteristic of a random event. And there is a statistical equation that can describe the distribution of virus particles per cell. It's called the Poisson distribution. This turns out to be the best way to describe this. So you add a certain number of particles to a cell culture. The Poisson distribution will tell you how many cells got how many particles. So let's go through this in the math. Here on the top is the Poisson equation. And it's pretty straightforward. pK is the fraction of cells infected by K virus particles. So if you wanted to know how many cells got 0, you would simply say P0. And E is the natural log, of course. M is the multiplicity of infection, which we've just talked about. K in all cases is the number of virus particles. So if you wanted to know how many uninfected cells are in a culture, all you have to do is take the natural log and bring it to the minus multiplicity of infection. And we'll calculate this out in the next slide. Very simple, straightforward. If you want to know how many cells get 1 particle, P1 is M times E to the minus M. So you simply multiply E to the minus M times the multiplicity. And if you want to know how many cells get more than 1 virus particle, that is 1 minus E to the minus M times M plus 1. And what this is, we're subtracting from 1 the sum of all probabilities for any value of K, the probabilities of P0 and P1. So that's P0 and P1, right? So we're just simply taking 1 and subtracting how many get 0 and how many get 1 particle. So that gives us more than 1. So let's look at some examples of this. If you take 1,000,000 cells and infect them at an MOI of 10, and you use the previous equation and ask how many cells are uninfected, the answer is 45. 450 cells get 1 particle, and the rest receive more than 1. So an MOI of 10 is a really good way to infect the entire culture. In fact, you only need to do an MOI of 5, and that's sufficient, but we typically use 10 in virology, because you see very few cells out of the 1,000,000 are uninfected or just get 1 virus particle. 1,000,000 cells infected at an MOI of 1, 37% of the cells are uninfected. So here is a way to get at least a 2-step growth curve. 37% of the cells get one particle, and 26% get more than one. So the uninfected cells are 37%, and the rest are infected. And a million cells infected at MOI of 0.001, 99.9% of these cells are uninfected. But some are infected, 0.099% get one particle, which is 990, and 0.0001% get more than one particle. So this is a very, very low infection. It's not zero, but it's low, and you can do multi-step growth curves at an MOI of 0.001. So let's see if this all makes sense to you. Here's the question. If cells are infected at an MOI of 10, in the growth curve, what will you see? So we're doing a one-step growth curve. We're using an MOI of 10. What's the curve going to look like? 1, multiple bursts of virus release. 2, multiple eclipse periods. 3, a single burst of virus release. 4, no burst of virus release. And 5, asynchronous infection. So you might want to pause here before I give you the answer. You can even go back to the previous slide and remind yourself what happens at an MOI of 10. But the answer, of course, is you get a single burst of virus release. At an MOI of 10, nearly all the cells are infected. You'll get a one-cycle growth curve, so you have a single burst, which of course means one eclipse period, not multiple, and the others are all not correct. So we've talked so far about how to measure infectivity and how to manipulate it with multiplicity of infection in a single-step growth curve. What are ways to physically measure virus particle? Well, there are quite a few. We'll just talk about a few of them here today. There's an assay called hemagglutination, which depends on viruses sticking to red blood cells. You can take pictures of the viruses by electron microscopy and count them. Many viruses have enzymes in the virus particle, and you can develop an assay for the enzyme. You can use serological methods which employ antibodies to measure viral antigens, and there are nucleic acid-based approaches. So let's take a look at some of these. Hemagglutination was the first quantitative assay developed for animal viruses before the Plaque assay. And what you do here is you take a preparation of red blood cells and you add dilutions of viruses to them. And you're depending on the property that some viruses have of being able to attach to red blood cells. So in this case, the virus is influenza virus. And influenza virus attaches to a cell receptor, which is a sugar that happens to be present on red blood cells. And the virus does not replicate in red blood cells, but we're simply using them to measure the number of particles. If virus is present, the virus will bind to red blood cells, and then the red blood cells will stick to one another by virtue of the viruses bound to each other. So you get this lattice that's formed, and that's the basis for the assay. So let's see how it works. On the top we have two virus samples, C and D, and you see we've made twofold dilutions, starting at 1 to 4, going all the way out to 1 to 8,192. Then we mix the dilutions with the red blood cells, and we let them sit at room temperature for a while. You see the top sample, C, contains no virus. That's because all the red blood cells have tumbled to the bottom of the well, and they form this nice, tight dot. When virus is present, the dot is not formed. And you can see in sample D, these wells have a nice layer of red blood cells coating the well. There's no dot at the bottom. But at the dilution of 1,024, you can see a dot begins to form. So we would say the titer of this virus is 512 HA or hemagglutination units. So you simply pick the last dilution at which you see hemagglutination. So hemagglutination is the name for this property of viruses binding to cells and forming this nice coating of the well. Now you may be asking yourself why the first well in sample D has a nice button at its bottom. The answer is that there's so much virus here that the hemagglutination reaction has been reversed. It turns out that the virus has an enzyme in the particle that reverses this attachment reaction. It is a bit slow, but if you have a lot of virus present, it will happen very quickly, as it does at this low dilution, but you can see it's not yet given time to act in these other dilutions. If you incubated this plate for another half hour at room temperature, you would see buttons in all the wells, so you have to read it right away. This will become more clear to you later on when we study the nature of the influenza virus particle. You can measure enzyme activity in viruses. Many viruses do have some sort of enzyme in them. This is a retrovirus shown here. On the left is electron micrograph. We'll be talking a lot about these viruses in this course. On the right is a schematic diagram of the particle. And inside the particle, along with the nucleic acid, which is shown in green, there are several enzymes, one of which is called reverse transcriptase. And this is an enzyme that can take an RNA molecule and make a DNA copy of it. So this can be readily assayed in virus preparations. And what you do is simply permeabilize the particles with a little bit of detergent. You add substrates for the enzyme and ask if it will simply produce a product. This is often done with retroviruses because they don't form plaques typically in cells. So here's an example of such an enzyme. Here, 3 different cell lines, shown by the names at the top, have been infected with a retrovirus called XMRV, either undiluted or at 1 to 10, and there's also a mock infected lane here. And at different days after infection, 1, 2, 3, 4, a little bit of the cell culture supernatant is taken, and then it's subjected to an assay for the reverse transcriptase enzyme in the particle. You can see in the mock infected cells there is no dark color, which means the assay is negative. And here in the first set of lanes of wells, you can see a dark color, meaning that the reverse transcriptase assay is positive. And if you dilute it tenfold, you get less of a signal. So these cells, the LN cap cells, can be infected, but not the DU145. And the WIMPy1 cells can also be infected, but apparently not as well. They're not as good a producer as the LN cap cells. So measuring enzymes in virus particles is another way to measure the particles themselves. We can also use antibodies to measure virus particles. So on the left, we have a viral antigen. This could be in a clinical specimen or in a cell culture supernatant. And let's say you don't have any other assay, but you'd like to measure the protein itself. So if you have an antibody to this viral protein, you can design what's called an enzyme-linked immunosorbent assay, or ELISA, to measure the quantity of antigen. And what you do is you first attach your antibody to a plastic support, shown here as a solid support. Then you add, say, cell culture supernatant, which you think has viral proteins in it. And if there's viral antigen or protein present, it will bind to your antibody. You then add a second antibody, which reacts with a different part of the viral protein. And that second antibody has some kind of indicator on it, a fluorescent indicator or a chemical indicator, so you can measure its binding. So by adding that second antibody, you can find out whether there's antigen present or not in your sample. And this can be quantified to measure the quantity of antigen. Since we're on ELISA, I'm showing you on the right, using a slightly different form of this assay to actually measure antibodies instead of antigens. On the left, we're asking, is there any viral protein present? On the right, we're taking a clinical specimen, typically blood or serum, and asking whether antibodies to a virus are present. And what we do here, we attach viral antigen to the solid support, and then we add our sample. If our sample contains antibodies, they will attach to the viral antigen. And then we add a second antibody, anti-IgG, an antibody against the first antibody, which has an indicator. And that way we can measure the presence of antibodies. So it's a very powerful technique for measuring both viral proteins and antibodies against viral proteins. And we'll come back to this again often in this course. Another way to measure virus particles is by green fluorescent protein, originally found in jellyfish, shown at the left here. The green fluorescent protein is a wonderful protein, a cage-like protein with a fluorescent center, which glows green in the presence of ultraviolet light. And this, of course, was used here at Columbia, first in worms in C. elegans, to show that this protein was a good tracer. This protein, the gene encoding this protein, can be inserted into viruses, and those viruses, when they infect cells, will make the cells turn colors. Since the discovery of green fluorescent protein, it's been modified in many ways to make different colors. And here you can see this is actually a small part of a cell culture dish, which has been infected with a number of different herpes viruses that have different colored fluorescent proteins in them, including green and yellow and blue and red and so forth. And so you can trace virus infections by inserting these various fluorescent proteins. And on the left is an example of a neuron infected with a herpes virus that produces a green fluorescent protein. And you can see it beautifully labels the axons and dendrites as well as the neuronal cell body. In recent years, it's been possible to see single virus particles in infected cells under light microscopy conditions by using these fluorescent labels. On the right is an example of an experiment using green fluorescent protein labeled HIV type 1. And this particle in green that's boxed and shown with an arrow is a single HIV particle. This is a photograph taken under a light microscope, low power. You can see all the individual virus particles. This is made possible, of course, because the fluorescence amplifies the size of the particle so it can be visualized. And the red in this slide, these are actually virus particles that are inside of a cell. The red stains the microtubule network of the cell, and you can see on the lower panels the movement of the individual virus particles on the microtubules. It turns out that viruses do move in cells along these microtubule networks, and we'll talk about that later. Two nucleic acid techniques have revolutionized virology, and I want to talk very briefly about them. The first is polymerase chain reaction. And this is a way to take a double-stranded DNA molecule, shown at the top right, for example, and make many, many copies of it. So in other words, you can take a small amount of DNA that might be present in a clinical specimen and amplify it enough so that you can detect it very easily. The way this works is you take your double-stranded DNA, it could be viral or otherwise, you denature it, you now have two strands. You now anneal short primers to those strands, which will make a copy of each one in the presence of a DNA polymerase. So we've gone from 2 copies of the DNA, 2 strands, to 4. You now denature those and repeat the priming and DNA synthesis, and you have 8 strands. And you repeat it, and you have 16. You repeat it, you have 32. So you go through multiple cycles of denaturing, annealing, and elongation, and you have exponential growth of this product. So where you might start with trace amounts of DNA, you now have a ton of DNA that you can easily visualize. This has revolutionized research. It has applications in industry and disease diagnosis. And we'll talk a lot in this course later on, especially about how, using PCR, we can diagnose viral infections. The revolutionary part the other revolutionary part of this procedure is that it employs a DNA polymerase that is heat stable, because there are cycles of denaturing, annealing, and elongation. The denaturing happens at 100 degrees, and so these enzymes have to be stable. And it was made possible by initially the discovery of bacteria that grow in hot springs. Their DNA polymerases are stable at very high temperatures. And those enzymes, which were first discovered in the 60s and studied simply because they were interesting, in the 80s were used to develop the polymerase chain reaction. Another example of how many discoveries in science have no apparent use, but eventually can revolutionize the field. Another revolutionary approach has been deep high-throughput sequencing. Back in the 70s, a way to sequence the bases in DNA and RNA was developed. It's shown here in this photograph of a gel, and this shows the sequencing reactions run out on various lanes. And I used to do this kind of sequencing reaction. And perhaps in a given day I could sequence 500 or 1,000 bases. So it was very slow, although it was very powerful. Now we have new methods of sequencing which no longer involve these kinds of gels, but which can sequence millions and millions, hundreds of millions of bases a day. And not only many, many bases in a rapid time frame, but a single DNA strand can be sequenced many times, so you have confidence that the sequence you get is correct. That's where the deep sequencing comes from. And the high throughput comes from the fact that you can do millions and millions of bases in a day. In fact, there are now machines that do this, which can sequence an entire human genome, 3.2 billion bases in one day for under $1,000. It's really remarkable. This technique has brought about the age of metagenomics, where we can sequence all the nucleic acids in a specimen, whether it be a clinical specimen or an environmental specimen, the ocean or dirt or the surface of a rock. It enables us to identify new viruses and new pathogens, and we'll talk about this in this course. So here are two examples of this. These are two episodes of the podcast This Week in Virology, where we talked about new pathogen discovery using polymerase chain reaction and deep sequencing. The first involves a new virus of snakes. There was a snake disease called inclusion body disease that was known for some time, and these investigators took samples from the snakes and found a new virus in them by PCR and deep sequencing. And at the bottom, the identification of a new virus called Heartland virus from patients in Missouri with severe febrile illness. Again, patients presenting with symptoms, no known cause, after going through many diagnostic tests. You take a little sample of serum and you can find a new virus. And this virus has since gone on to infect others as well. So it's an incredibly powerful new set of techniques that we have to push virus discovery forward. However, they do not come without caveats. And I want to illustrate this caveat in this slide. A number of years ago, a paper was published called Zoonotic Viruses Associated with Illegally Imported Wildlife Products. Now, it turns out that many wildlife products are illegally brought into the U.S. from various countries in the world, and some of them are shown here on this slide. You can see this is a monkey skull and various parts of monkeys' limbs. And here on the right is a set of monkey limbs confiscated at the airport in Chicago. So it's illegal to bring these in. These are dried and not infectious if there are viruses in them, but it's illegal to import them because this is wildlife material. So they're confiscated often at airports in Chicago and New York. And in the study on the left, which was carried out by a group at Columbia Medical Center, they used PCR and deep sequencing to ask, are there any viral sequences in these specimens? And they did discover a number of different virus sequences. And in fact, the New York Times picked up this story in January 2012, From the Jungle to JFK, Viruses Cross Borders in Monkey Meat. What I want to caution you about, though, is confusing viruses with viral sequences. I've just told you how polymerase chain reaction, PCR, and deep sequencing can allow you to discover new viruses. But you can't simply say a virus is present if you have a part of its sequence. You have to actually find the virus. And what they did with these imported wildlife products is they used PCR and deep sequencing and found viral sequences, but they didn't find virus. So to say viruses are associated with these products, both in the journal article and in the Times, isn't really quite correct. The correct thing to say would be viral sequences or viral genomes are found. And if you really want to prove that a virus is present, you have to go further than the sequence. So the sequence gives you a clue as to what might be there, but you have to either isolate infectious virus, or if you have a living animal, show that there are antibodies to it. If you could identify the entire viral genome from multiple specimens, that would be good as well if you can't isolate the virus. But I want to just caution you at confusing viruses with viral sequences.